All right, today Shachak, our own Shachak, is presenting. He received his PhD in geophysics from the Tel Aviv University in Israel. 2005, he came to UNH here at CCOM and did a postdoc fellowship with the Tycho Award. He's currently a research professor here. His research interests are the optical remote sensing in the littoral zone, with a focus on experimental and theoretical studies of LiDAR remote sensing, which includes airborne LiDAR bathymetry, topographic LiDAR, and terrestrial laser scanning. He also is into hyperspectral remote sensing and sensor fusion.
Thanks. And now also into biology. So, this.
Not a bad thing.
Actually, I'll come to that. So, the original talk was supposed to be given by Dr. Gidon Tibor, who is actually listening to this broadcast. And if, Gidon, you have anything you're interested to say, please text into the chat box and they'll unmute you. So, unfortunately, Gidon could not have been here, and instead I drafted up a different presentation and incorporated some of his stuff. Now, a common thing between Gidon and I, we're both geophysicists. We actually had the same professor, Dr. Tzvi Ben Avraham. And as a geophysicist, give me a hammer and hear me refract. I know how to measure tomography. I know how to map. My best friends are geologists that have a smaller hammer, and geodesists that have even a smaller hammer to get benchmarks. But I never actually worked with biologists. And this was quite an interesting experience. So that was a learning curve for me. And Jen was one of the people introduced me to the world of biology and ecology, which is different because I never heard about a term called genotype. For me, when you measure a shoreline, it doesn't matter how the shoreline looks like. You measure the shoreline. But here it can be different types of shoreline, different types of measurements. So those are the things that I will talk about. But before anything else, if you notice, I have a slight accent. This is because I come from a different location than New Hampshire. I'm originally from Israel. For those that don't know where Israel is, you might have heard it about the news. It's a very exciting place. Earthquakes all the time, and there's other kind of activity. And Israel is located in the Levant area. We put L in ISO. So for those that don't know, it's on the eastern part of the Levant. Sorry, the eastern part of the Mediterranean, because we'll see it from a marine perspective. And the Mediterranean is considered a warm, salty ocean. But if you think about the Israeli coastline, this is actually one of the more warmer places you can find the Mediterranean, and even one of the saltiest places you can find the Mediterranean. So the PSU, or the different terminology for that, the percentage over here is about close to almost 4% salinity at that location, and it's actually quite warm temperatures, around 24 degrees. And Israel has more than one water body. It actually has 4 water bodies surrounding it. And it might be a little misleading thinking from the Bible terms, the Sea of Galilee. The Sea of Galilee is actually quite small. It's smaller than Squamscott, actually. And this is actually a freshwater lake. And the temperatures vary quite a bit, because the temperatures in Israel vary quite a bit. And it goes from 14 degrees centigrade to 30 degrees centigrade. And it's considered one of the coldest water bodies. The Mediterranean, as I mentioned before, is 22 degrees centigrade with a salinity of 39%. This is, if you compare it to the Atlantic Ocean, which we have here the Gulf Current and the saline water coming from the Gulf Coast, is even more saline than the Gulf of Maine salinity. But the most impressive place is the Dead Sea, where the salinity there gets to 342, which is quite salty. It's one of the saltiest places you can find. Temperature-wise, it's still in the nice moderate temperatures of 22 degrees centigrade. But my favorite is the Red Sea. The Red Sea is what I call an average normal temperature of 26 degrees, which is 79. For New Hampshires and Maines, that's bath water, but I love it. And the salinity there is about 41 PSU. And the National Center in Israel, the National Center that researches that is called the Institute of Oceanography and Limnology Research. So before going into the presentation, I'll give you a little review about the center. And in addition, I'll talk about the new purchase of a Bat Galim research vessel, and that was the original presentation. And then I'll go into the coral research that I'll present today. So I'll start with the IOLR. So the Institute of Oceanography and Limnology Research actually has 3 centers. One in Haifa, which is the headquarters, and that's what's called the National Institute of Oceanography. And 2 other centers that are located near the Sea of Galilee, near Tiberias, the Kinneret Limnology Laboratory, and one near Eilat called the National Center for Mariculture. And I actually asked, what does mariculture mean? And from what I understand, it's aquaculture, but also studying some algae in the water, so that has a different term to it. So that was quite interesting, and that is located in Eilat. Similar to the Joint Hydrographic Center, CCOM, IOLR has 2 hats also. So that was the government hat. The academic hat is in collaboration with the University of Haifa. And it's under this research center called MERCI, which means thank you in French, is utilizing this research institute to investigate technology and mainly ecological features. Recently, they discovered gas, actually natural gas, in the Mediterranean. So a large amount of money has been awarded to the University of Haifa, and they're now purchasing new equipment to monitor and evaluate these different ecological features, that I'll say shortly. And those interests follow from its engineering perspective, from port expansion and artificial island construction, to more monitoring environments such as desalination plants, which we have in Israel 4. It's actually one of the biggest density of those. Marine outfalls and brines that can come from these desalination plants. Mariculture and aquaculture development. And now also gas drilling and the increased transportation. In addition to that, we are trying to even build more collaboration between Israel and Jordan, and maybe it's beyond the Israeli side, having a conveyor belt, an inland channel between the Red Sea and the Dead Sea, to have a water coming through and maybe filling up the Dead Sea or having some transportation considerations there. The issues that are of interest to the IOLR are the same interest to NOAA and other national organizations here in the US, from the public health and water pollution to coastal erosion. The coastal area of Israel is mainly sandy and sandstone. So it's carbonite and silica. And although here you can see that other people are bathing along the shoreline, this shoreline is starting to erode, and sandstones are falling over time, and then they're getting washed away, and this procedure happens again. In addition, overfishing, this is a whole Mediterranean issue and an international issue, is something that is a big problem. This resource is starting to disappear. And in addition to disappearing resources, well, a big invasion from the Red Sea through the Suez Canal that is starting to affect the Mediterranean. And there's a different kind of fauna, such as lobsters and jellyfish. Jellyfish was yesterday in a conference, and I heard that it's not a problem only for the Mediterranean, but also for the north part of the Atlantic, where around Belgium and England there's more and more jellyfish appearing. Last but not least are the coral reefs, which any changes in the environment, they're like the canary in the cave. An impact on the environment, they will kind of indicate that, and that's what we'll talk about. There are much more things that are happening in IOLR. I won't go into the details about it, but if you're interested, please visit the ocean.org.il website, and you can learn much more about their activities. But the main thing that is new in their activities is the collaboration with CCOM. Last year, Dr. Thibault came over here and came as a visiting scholar, and through an agreement, a memorandum of understanding, we have now a collaboration between JHC and IOLR. After his visit here, I actually was in Israel, and during my research there, I also met with the scientific attaché of the US Embassy, talking about collaboration. And and we decided to work on two projects. One is monitoring the northern coastline of Israel, and the other one is spectral characterization of corals. In addition to all this collaboration, Gidon Tibor et al. Started to consult with our center about building a new ship, and because we have expertise here, how to incorporate and meet the standards that are required to do good surveying and good mapping. I will focus here, as I mentioned, mainly on the coral research, but just to put a note out there, if anybody is interested on the other study, the seafloor characterization, there's a poster on the second floor that I just put, and you can go through and read about it, and I'm more than happy to talk offline about this study as well. So let's talk about the ship that was bought by IOLR. So what happened was that the young lady that was 51 years old, the Shikmona, was a little out of date. The biggest challenge is the operating crew was only 5 people, and the science crew was only 15, at most 15 people. So that was a little challenging. It was also limited how far it can go, overnight operations. And it's been operation in IOLR since the 1980s. So leave it to Gidon to find a better ship to replace the Shikmona. Luckily enough, they found this small class, the Kleine class, from the German Navy, that after they did their torpedo experiment, they had 6 ships of those available, which is quite nice, very quiet vessels. They gave one of his ships to the Israeli Navy, and the Israeli Navy, at 2008, I think, and then the IOLR purchased it this year, which is ideal for hydrographic mapping. One of the modifications that are done on this vessel is to bring the crew capacity from 14 to 26 to allow more people on board and conduct research much in a broader scope on this vessel. This is, I don't know, I don't find it interesting. One of the interesting points to mention about the vessel is the goal was to meet the highest standards possible for hydrographic surveying. And this was done with the collaboration of Tim Gates et al. And what they tested out is to see that the acoustic noise level on this vessel is good enough to yield full sonar performance. In addition, they also checked out the noise occurring from the bubbles, and they noticed that is not a significant factor to affect the measurements. And Gidon wanted me to emphasize thanking the CCOM people that were involved, Larry, Tim, and JB, helping and consult and construct modify the vessel according to hydrographic specifications. One thing of interest on the vessel was the gondola. Now they are adding to the hull of the ship a gondola which will allow to put a whole suite of acoustic sensors and conduct measurements. And this is including ultra-short baseline transmitters, hydrophones, a multi-beam, and I think there's a few other things that are Knudsen, I can see here. There's a few kind of sensors that are putting here that we're planning to conduct all kinds of surveying with. What I found very interesting was their A-frame. They're actually building an A-frame, which is about, I think, $200,000, that is telescopic, that can extend from 4 meters to 7 meters, and actually can work in sea state 5, which is when you have whitecaps and a very gushy ocean, with 5 tons on it. So that's quite impressive. Without going into too much detail about the different sensors but on board, it's actually quite nice to see how they identified for different types of activities that require different sensors to operate, for mapping the seafloor, for sampling the water column, and even for emergency response such as oil spill, but now that there's natural gas and other kind of exploration in the Mediterranean, this is as important as monitoring the environment. Another point that I saw quite interesting on the bat galim that they're adding is they're planning to add an ROV and an AUV based on the HUI model, so like the JSON from the Woods Hole Institute of Oceanography, and a Remus 3000. This will be purchased via the University of Haifa, and that will be part of operation that is going to happen on the new Bat Galim ship. So that was quite of interest and allows us to do some comparison to some research and interest that CCOM has in it also. So that's more or less my review about IOLR. And now we'll get into the more interesting stuff, which is the habitat mapping. So this research started while I was visiting Israel. And when I was going through the different laboratories, I was talking with a chief researcher that was involved and asking what's their interest and what their challenges. And one of the most interesting people I met there was Professor Bukirinkevich, who is very famous in his field, who investigates corals and specialized specifically in this type of species of coral. It's called Stylophora pistillata. And that's one of the common coral species, almost as common as another type of species called Acrophora. And the interesting thing about it, it actually had some characteristics that I could identify and spectrally characterize, because Bukirinkevich already conducted some laboratory work, so we know what's going inside this kind of species. And then when I looked at my measurements, I could identify and characterize it. Now, my biggest problem is, I told him, is I'm a remote sensing guy. I know how to take a sensor, such as a hyperspectral sensor, which collects imagery on a broad spectrum of light and samples them at a very narrow bands, every 10 nanometers. And then I get a signature, which gives me a color characteristics of a feature I'm measuring. For example, this is vegetation, where you can see the chlorophyll pigment, and I'll mention that quite a few times, which you can see a small peak here in the green and a very high peak in the infrared. And I know when to see that when I have it underwater, when it's attenuated by light, and then most of the characteristics are in the 400 to 600 nanometers. But having that and trying to characterize that, that's a whole different story. One of the collaborations that I have conducted for the years was with NCOS at NOAA, with Tim Batista group, the biogeography group, where we went around, collected with hyperspectral imagery, images over different habitats, namely coral habitats, and try to characterize them and separate them. Now, for me, as a geophysicist, I see here only two types of features. I see here some green stuff over sand, some coral, which is like more carbonite stuff, some green stuff or brown stuff over sand, and sand. From a biologist's perspective, I said, oh my God, what are you talking about? This is seagrass. Can't you see the beauty here? This is hard bottom with with some algae on it. This is macroalgae. And yeah, this is sand. So that's more or less the kind of the characteristics. And I was able, based on this kind of interaction, to characterize and classify this seafloor and provide a map. And this follows a much more complex kind of map and habitat classification.
Chuck, you take a step back and tell us what you based that classification on?
This actually, it's based on this on the NCOS classification scheme.
You know, what parameters in the local sense data? How did you differentiate?
The spectral characteristics that I use to characterize and classify these features are namely in the blue-green region, between 400 to 600 nanometers, because water attenuates things that are in the infrared, which is typically what you can identify as vegetation. Based on these kind of characteristics, I first correct for the water column, remove attenuation of the water, and then I look at the color that I can measure, and then I conduct a beginning unsupervised classification to see what are classes, compare that to the diving sites, because NOAA likes to conduct monitoring also in situ, so there's diving sites that come and visit such as the macroalgae here, the seagrass here, and tweaked my algorithms accordingly by getting a signature, meaning a characteristics that I can use to guide my classification, and based on that I have a supervised classification that I classify the seafloor. More answer to the question? The important thing here that there's a much more complex classification scheme that is in place. And in order to do that, we use actually the whole suite of surveying systems that we have. Typically for the morphology, which goes beyond the optic level, and in Caribbean water it's very clear, you can go easily 50 and 70 meters with airborne lidar bathymetry. With hyperspectral only 20 meters, but with multi-beam echo sounder you can map all the way through, and then you can start mapping that the general bathymetry and giving that some attributes. Then, after that, you can start looking in a little more finer scale and starting to characterize all kinds of patches and types of rubble and any kind of other kind of seafloor characteristics to put more attributes. For that, in addition to the bathymetry mapped by the airborne lidar bathymetry and multi-beam echo sounder, I use hyperspectral imagery to get that color characteristics that I can use to classify and characterize that. The benefits also using hyperspectral imagery, if I have mixing, meaning two types of features in the same place, such as sand and vegetation, I can also use that to estimate patchiness and mixing. More challenging, but still possible, and this is more on the research level than operational, is trying to identify biological cover. And typically, it's always assumed that we're talking about live coral. It's important to identify that, and typically it's identified like you identify vegetation. Does it have chlorophyll, which is a green pigment that identifies. But if you don't have it, then it's a problem. But that's where it stops, and the question is, maybe before going into that, there's another aspect we need to look at, and that is the health of a coral. Now, that was an interesting question. I said, I'm not so familiar with coral health. How can we look at that? How can we investigate what's going on? So in order to do that and to bring you up to speed, how to conduct so this is like do-it-yourself kind of spectral measurements. So if you can find a coral, I'll just give you the instructions how to do it in your own home. The only things you need are some basic radiometry, some coral biology knowledge, and a bit of ecological knowledge, and you're all set. So let me run it by you quickly. First, a little bit of radiometry, some background. In order to conduct spectral measurements, I need a light source. The light source will shine over a target that I have, and what I'll do is I'll use a detector to collect that energy coming back. If I have a broad spectrum of light, so let's say from the ultraviolet to the infrared, I'm transmitting it over a coral species. I'm looking at the data that's collected and I'm normalizing it. I will get a signature that is called reflectance. And here you can see a reflectance signature where I get some green occurring here. There's an absorption peak in the 670 nanometers. And then I have a strong reflection in the infrared. I can do actually the same kind of experiment with a narrow beam, such as a laser or a blue underwater torchlight, which is in the 425 to 475 nanometer. And this is actually quite a useful tool, because in addition to get the same kind of reflectance characteristics that I mentioned in the top part, I actually can get here also emitted energy. And what is the most common emitted energy that you can get from vegetation? Fluorescence. There's other kind of scattering that are occurring. It's called inelastic scattering, which you have energy transmitted at a different wavelength than the one you transmitted from. And you started to see other characteristics here, like you can see here the reddish hue on top of a coral. But he's talking about some different characteristics going on here, and that allows us to evaluate what's going on. So now I have two types of different characteristics that I can use and analyze with data sets. Now, it's easier to look at it from my perspective in equations when I have a transmitted radiance, which is watts per square meter per steradian, and I'm looking at the energy transmitted going down and the energy reflected back going up, and I'm looking at that ratio. And if I'm looking beyond the spectrum that I'm reflecting back, I can look at the fluorescence component. And based on those kind of radiometric parameters, I can now conduct my calculations. So now that you all know radiometry, we can talk about the coral tissue. The coral tissue for me was a very hard thing to grasp because, again, I'm a geophysicist. When I look at this, I can see stratigraphy, I can see geological layers. And that's how I looked at it. And an oceanographer can look at it as sound speed velocity changing in the water, which is another kind of analogous. If I shine light through the coral tissue, I have light transmitted, reflected, and refracted through these sublayers, which going through this mucus layer, through the ectodermis, muscoglia, and I won't pronounce the other things but to make less mistakes. And when it goes through all the way to the bottom, it interacts with a coral skeleton, which is another aragonite skeleton, carbon calcium, which reflects back all this energy, because carbon calcium provides like a Lambertian scattering, reflection light in all directions. And then I can collect this light. And I can measure also the coral tissue without the coral skeleton if I can get a larvae sample. A larvae is called planula, which are found within the coral branch. And this is this tissue that is like flipped, and you can look at it as a flipped tissue that you have a mucus on both sides, and you have light transmitted going through and then reflected back. In addition to the light transmitting back and forth, we also have some algae, symbiotic algae growing within the coral tissue. It's called zooxanthella. This has a pigment, this species have a pigment that's called a chlorophyll pigment that creates that fluorescence that I mentioned earlier. So I have this energy also reflecting back and collected, and also collected for analysis. And when I looked at that, the first thing I thought about was airborne light dosimetry. So when I work with airborne light dosimetry, I have a laser transmitting through the water surface, interacting through that, going through the water column, interacting through all the scattering, interacting with the seafloor, and then reflecting back up again. And when I collect that, I can actually create a time series and measure the different kind of properties that I have within the water column. There's only one little problem. The water bodies that I'm looking at are typically in the orders of meters. And light, you can't go beyond the speed of light. 1 nanosecond is equal to 30 centimeters in air, which is 1 foot. In water, it's 22.5 centimeters. So in order to do a similar thing with coral tissue, I need to find some LiDAR that works on a picosecond rather than a nanosecond level, which is unrealistic. Instead, when I'm looking at light transmitted through the coral, onto the coral tissue, through that whole transparent tissue and back into the detector, I can look at it for the spectral range, meaning instead of time, I'm looking at wavelength. And the key issue, I can look at the energy that is reflected back, which is from the scattering, the elastic scattering, and I can look at all the phenomena that are occurring outside the wavelength I transmitted. And there's quite a few mechanisms that are considered inelastic scattering. I will focus mainly on fluorescence, but another interesting property that is worth investigating is Raman scattering, which is related to the medium that you're transmitting through. But looking at the pigments, those color that provide the energy back at a different wavelength, so they carry it from, let's say, the green and transmit it in the red. Those are the pigments that I'm interested to identify, because beyond chlorophyll, there's protein pigments that indicate other kinds of situations. So after we covered the coral tissue, last but not least is thinking about stress and disease, very happy topic. How we can think about it is similar to thinking about ourselves. For me, it's a little harder connotation, but imagine I have a lot of hair. Now I get stressed, I think about all these problems, I can lose color, I can gray hair. Even worse, I lose all my hair. Corals are not different than me. When you stress them out, they start to expose algae. They need to maintain some balance. Now this algae is very important, this zooxanthella. This algae is the one that's managing the light coming into the coral. Too much light is bad for the coral. Too little light is too bad for the coral. So these algae are used as valves by first taking light in and absorbing it. And if there's too much light, they are actually emitting it out, like we talked before, fluorescence. Any change in the ocean conditions, such as light, temperature, or even other properties such as ocean acidification, will affect that and will cause stress to the corals. The most common phenomenon that is noticed is coral bleaching. And there are several papers about that, what exactly is the cause and how it is occurring. And if it is a death sentence to the corals. And that's a big discussion that we won't go into. Worst case scenario is after the coral is not able to maintain these zooxanthellae and cannot maintain this light budget, it starts to die. It has diseases, and there will be parts of the coral that will die off, not all the coral, and these areas will be a good bed for bacteria and other kind of things to grow, and that's what is typically called a diseased coral branch. So, after having all this knowledge, let's start giving a literature review here. And although talking about biology is important, I thought it was important to also mention a seafloor mapping effort that was done on a biblical proportion. Moses et al. Were the first to map the seafloor. They actually did it very bad, wasting of money. Let's just going like that. Unfortunately, not a lot of documentation. I mean, actually, talking about documentation, like a letter to nature, this has the biggest impact factor. But but more important papers that were written about coral tissue focused mainly on the skeleton because of its reflectance characteristics. You have all this energy coming back. And using the tissue as a filter, and you try to indicate the health of a coral based on the reflectance from a coral. Other studies focused on the chlorophyll concentration based on this zooxanthella that I mentioned before, and looking when it gets lost, so having less fluorescence occurring, and that was the indicator. Now, these are important properties. However, as I mentioned before, to answer that question, this focuses only on the skeleton, and the wavelengths of a chlorophyll are focusing mainly on the infrared, 735 nanometers and 685 nanometers, which is close to the infrared, which are absorbed quite quickly in the water column. So we looked at areas that are in the more blue and green regions of the light. And when we tried to figure out where are we going to sample, we needed to take a bigger overview of this area. So today, the coral beds are found in the shallow waters up from 0 to 4 meters of water, and actually it's all around these areas where typically you can't go and survey with a vessel, so those are areas with gaps. But there are other coral reefs that are noticed that are more ancient in the deeper waters. Now the interesting thing here, and there's a study by Mo Canary, noticed that there's sediments always coming when there's great floods from the desert area down from the south to north, and that chokes some of the coral beds, and that's what noticing here about the ancient corals. They don't have enough light when you have so many suspended particulates in the water. And this you can see here, so these ancient reefs, some of them are completely fossilized, some of them are partially living, you can say. And that's what's interesting. And this is in an area that is also tectonically very much active. This is a left lateral strike slip for those that are interested. And it's the last biggest earthquake was in '95, I think, the Nueva earthquake, which caused here quite a bit of damage. For our research, we focused on two areas that we know we had healthy corals. They were in two different depths, just to see the effect of the ambient light. One was at 5 meters of water depth, and one was at 12 meters of water depth, and we were quite lucky. The period that I was in Israel, which was end of May, beginning of July, that was that's when the corals are very productive, very fertile. So we're able to get healthy and sick branches from site A here. But in addition to getting these branches, we found very fertile branches that we could extract from the larvae, planula, to investigate. After conducting this research, we were able to validate that by revisiting this site and measuring in situ these corals and seeing if we were getting similar results. Luckily for me, IOLR has a darkroom where they typically conduct coral research using UV light. So using some of Gidon Tibor's equipment, I was able to configure quite quickly a spectral laboratory. And for those of biologists will notice there's all kinds of biological thingies that I don't know their names, and I just place them around, and they became my bed and my sampling areas. Used Gidon Tibor's spectrometer, and where we use the fiber optic connected to the spectrometer, where we measure the sample. We calibrated using a spectralon, and then we measured the samples. Here you can see a picture. I hope you can see the picture of a fluorescence that we're measuring. Similarly, in the ocean, we conducted the same kind of measurement. Now, these, just to disclose, this is from a study that was conducted earlier by Tim Batista and I in the Virgin Islands. I just didn't have pictures of the fieldwork there. What we used there was a similar configuration. We had a laptop, a spectrometer. Instead of using an underwater camera, instead of using an underwater video camera, we used a diver camera. But we used a fiber optic, 25-meter fiber optic cable that went down with the divers to collect the spectra in situ from the different corals that the biologists identified. And all these were logged and measured, where we had an image giving us the texture of the coral and a spectral response that we can use to characterize. What I'll show you now, and this is the benefit of presenting at CCOM, I know I'll get honest feedback. Some of this is also really good, and we are ready to publish it. And that's why I want to hear your feedback. This, and in addition to that, we have some additional results that are similar to the chlorophyll studies that I mentioned before. And if you have feedback about that, I'm also very happy to hear. So, as I mentioned before, the reflectance characteristics is the measurement of the ratio between the transmitted light and the reflected light. And when we look at corals, we can, for simplicity, we'll break it to two parts. The green-red zone, which is 525 to 675 nanometers, and the red infrared zone, which is 675 and above. One of the things that I looked at when I looked at this reflectance signature, I wanted to see how well does it correlate with the chlorophyll absorption signature. So as I mentioned, corals have different proteins and different pigments. So I took the most common pigment and looked at the absorption spectra, and I noticed there are some differences going on. Namely, around the 525 to 575 nanometer, so that the tip of the green, it was at the tip of the green towards the blue. Using that, we started to conduct measurements on the coral branches, and we used an approach that's called base-to-tip approach, where we take samples along the base, and we compare them to edges which potentially can grow for the coral. They're called tips. Now, one of the things that I'll show you later on is in corals, and that's why I hate biology, I always think tips are at the edge. Sometimes, apparently, tips can go from branches in different locations. I hate biology. Anyway, using that, you can actually analyze and characterize what's going on. And based on these measurements, we can start to compare and look at the results. So we have base and tips, and we can index them and relate them to get some characteristics on this coral health. And for example, if we look at a healthy branch and we compare the tips measurements to the base measurements, we notice a clear difference, especially in this range that I mentioned. You can see this bump here that is missing there. And the slope, you also notice the difference, but this is very hard to characterize. You notice that the tip, the inflection point of the tip starts earlier than the inflection points on the base lines. So looking at the reflectance measurement, we said it's actually quite hard to analyze and make it a little more robust for indexing. So instead, we created the first derivative of the spectral signatures and looked at the slope response. And this actually was quite helpful, because now I can characterize the waveforms much easily. For example, in the red infrared, I can see that the peak of a tip much more clearly compared to the tips of the base measurements. And when I look at the green-red area, we notice another change between the tip and base, especially around the 580 to 600 nanometer, where we can see here a slope. And for me as a remote sensing guy, this is the holy grail. I can actually use that to create an index. I can take measurement either from the peak at 550 nanometers or 600 nanometers and compare them to that 580, 590 nanometers. So I can create an index here to use for classification. Alternatively, I can find the peak here and use that also for mapping classification. The main and most interesting results were found in the green and red, where the first thing we proved to ourselves that this phenomena that we see is not related to the chlorophyll. So as I mentioned, all these previous studies looked at the chlorophyll, and it's very true about the red and infrared. However, looking at this area, this is independent. And one might say, wait a second, I know Acrophora, which is a different species than that, and Acrophora doesn't have zooxanthella on the tips. Well, that's correct for Acrophora. For Stylophora, there is zooxanthella on the tips, meaning there is green pigments at the tips. So it's maybe related to concentration, but there's other thing going on here, because otherwise I would get the same kind of characteristics because of that pigment. So when I compare the three genotypes that I have here, these three species types, Jen, correct me if I'm saying something wrong, then you can see that there is correlation, not always the same, as I mentioned, genotypes, everything is a little different. Sometimes it's much more acute and clear. Sometimes it's a little more subtle, as you notice in genotype 4 and 5. And interestingly enough, genotype 3 didn't match the pattern. And in hindsight, and this is winning a punchline for later on, we discovered that this branch was actually sampled under stressed conditions. So we already started with a branch that was stressed, which we considered originally as a healthy branch. But it was interesting to have that as a control point. We also looked at disease branches. Remember, as I mentioned, there are areas in the coral, in some corals, that is dead, and bacteria grows on it. Now, bacteria is not it's not one species. It can be a whole diversity of stuff. And some bacteria, when you look at their spectral response, have a peak somewhere in the 580, and some bacteria have no peak, and it's like a flat, broad hill, let's call it, peak going all around. So this clearly cannot be used as a characterization. And when we look at base to tip measurements going from the diseased area all the way to the tip, it was quite interesting because we can actually see that we notice that all the point measurements here are under some stress conditions, because we're noticing this slope that I mentioned. But if we take it from the tip at 560 and looking at 585, we notice that the measurements are going downhill. Here it's much more clear, but this stressed out all the genotype 8, this branch. And to prove that point, we actually used another part of the IOLR facilities, and we used the coral laboratory, where they have tanks, small water tanks, aquarium, where they can put corals and measure that. So we put one type of coral in each of those tanks, lowered the temperature, and what's the thing I like about corals, they're like Israelis, from 26 degrees to 22 degrees centigrade, which is freezing for me, but still 70s for you, and see what's going on over time. And the thing that was interesting, in areas that were close to the base, we didn't see so much change. But when we looked at the tips, we actually noticed that even after 1 week, we noticed some stress occurring, and you can notice the slope is occurring at the tips of a coral. We looked at it on the other samples, and I'll go here quickly, but you can see the same thing. When we look at the tips, we notice the stress is occurring, and here's another genotype. Genotype 4, the same thing occurring again, and genotype 5. Sometimes there are other kind of phenomena that are not exactly following this second peak, like in point F has much broader kind of characteristics, but still it has this slope. And when measuring it, and I apologize, it was in a rush, I didn't put that index value between the peak at 570 to the 585. You actually can see it as a negative positive indicator, which is quite nice to use. And based on these results, we actually are quite happy, because we are able to produce an approach that is using sampling from the base to the tip that can characterize and monitor coral branches. Now this is a thing that hasn't been used so much now. It hasn't been used, period. And what we notice is that it's independent of the chlorophyll pigments that are in the coral tissue. So it's a nice indicator of other things that are going on. And again, our goal here is to try to figure out what's the next steps. Are there any fat layers or anything else? So we're using this empirical measurement to start guessing what's going on when we have this stochastic model. One light transmitted through all this layer, come back, and now we're trying to decompose that. And we noticed that the area that we found the biggest success for the Stylophora pistillata was between 550 to 600 nanometers. So that was quite a success for our perspective, and we're now working to publish these results. If you have any comments before we submit it, I'll be more than happy to talk about this more offline. The second part of the measurements we conducted was on the more conventional measurement, which is focused on the chlorophyll. And that was quite interesting because this, we could look at it from the branches, from the planula, and conduct some field measurements on that. So now we're focusing on this area where we see this red edge effect. You see how the energy shoots up and almost saturates after 700 nanometers. And here, similar to what I mentioned earlier, you can notice that the tip measurements occur before the base measurements. So the tip measurements are typically 3 to 5 nanometers less than the peaks you would see at the base measurements, which is quite nice. And that gives me a good feeling if I want to configure some spectrometer or some spectral measurements, how to separate between these conditions. And when we look at disease branches, this was quite interesting. We notice an inversion that is occurring here, that if you look here, you notice that the peak in the tips is lower than the peaks in the base. In the diseased, we notice that the base are lower than the peaks in the tips. So that was an interesting phenomenon to notice, that this inversion that is occurring. More measurements are needed to talk about the diseased branches. It's hard to extrapolate from only 2 branches about this kind of phenomenon. But we saw it also over time, and unfortunately I don't have the results here. The other property we're looking at was how the coral tissue affects through the planula, for these coral larvae, which have no skeleton, so we don't have reflectance, only internal reflectance from the coral tissue. And we could see that different planulas from the same branch were varying a bit between themselves, and you can notice not all of them have the same shape around the 520 to 560 nanometers, but in general, all of them have a similar kind of trend, this jagged thing that goes peak at 550, and a low at 570, and another peak at about 590, and another low at 610. And when you compared between all, it was hard to say that we're seeing any distinction, any one species belonging to one branch compared to another planula group belonging to another branch. Same for the infrared. The results were very similar. So the chlorophyll was the dominant factor, and we got the same kind of signatures with all of them. So there was no discrimination on this kind of level. However, when we looked at the fluorescence, this was quite interesting. So what is fluorescence? As I mentioned before, it's energy that is scattered back from a wavelength that is different than the energy we transmitted. An important comment to make here, this energy that is scattered back is typically 2 to 3 orders of magnitude smaller. So if the relative units that I'm measuring here are in the thousands, here I measure in the single digit or in the lower tens of intensity values. And what I typically try to look in chlorophyll fluorescence, for example, is these 2 peaks that have been mentioned quite a bit, and Anatoly Gitelson is the leading researcher on this phenomena, where you have a peak at 685 nanometers and a peak at 735 nanometer. And based on these 2 peaks, if we normalize it with respect to the background noise, we can actually index them and see what's going on. And looking at these coral branches from a fluorescence perspective, you can use that index calculation. And here what I did is calculate the peak ratio between the 685 nanometer to the 735 nanometer. And for a base tip, which is rich with zooxanthella, you get a high value, a 0.5 value of index. And when you're looking at a tip that has very little zooxanthella, but still has chlorophyll fluorescence, you get a 0.2. You don't get a 0, you get a 0.2. And this was quite consistent with most of the coral branches. You can see what's going on. In genotype 5, we noticed that we had some confusion going on, and when we looked closely at that, we noticed that some of these places that we assumed to be base were actually tips, because there were growing points in the coral branch. When we went out into the field and we measured that, and the H's here, I apologize, H1 to H3 are site A, the one at shallow waters 5 meters, and H4 to H5 are in the deep water, we actually see the same phenomenon, where the base that is marked with the letter B is the peak at 735 is always higher than the tip, which is marked with the letter T. So even if the energy is lower, we still see that ratio occurring. So it's a nice index that you can use to characterize the corals, especially discriminate between tips and bases. And again, an interesting phenomenon when we looked at the diseased branches. So we have this inversion occurring, where now the base are lower than the tips when you have a diseased branch. And at the moment, we haven't looked into too much. And the next step from a biological perspective, and again, I'm always looking at Jen to correct me if I'm saying something wrong, histology. We're now planning to cut it, which is shocking. Biologists work with a knife and dissect the coral and try to look for the layers.
That's what's great about this study, though, right? Because you can use spectral signatures, which is the first time that anybody's done that, as a measurement of physiological stress. It's a very instant gratification type of measurement.
And I'll repeat it for the audience listening. So the beauty of it, and those that are listening to me and don't know that I'm sarcastic sometimes, the beauty of this study is we're using traditional biological methods, where we're first using a reconnaissance by a spectral measurement, a non-invasive kind of measurement, and then we use an invasive traditional biological measurement where we can cut and look into the coral tissue, which typically in an invasive measurement, you damage the coral tissue. Schrodinger's law, you cut it, you compress it, and if there was cavities, you lose it, but you can still say what's going on there, but you might be losing stuff. So having an independent measurement that is not invasive is a nice complementary method that can be used. And same thing, looking over stress and time, we notice the decline of different points along the coral species, and I forgot to mention before, you can see those red dots that are pointing to the measurement, if it's a base or a tip, and you can notice that the interesting part is that the tips show the most change occurring under stress, and sometimes it's over a week, sometimes it actually takes about a whole month until they feel the stress, so it's completely different than the measurement before, where all the measurements showed a week of change. Here, sometimes it took a month until we noticed some change in the chlorophyll. Just to time, I'll go quickly over that and conclude that this study shows is a promising study that hopefully will get more samples and can have some publishable results here. It's very interesting, and we actually are getting collaboration with NCOS to see how we can apply that and incorporate a collaboration between UNH and NOAA. But still, in order to have a really good study, more samples are needed and more work is needed to present that. Yes, Colin?
Yeah, so you were comparing measurements in the lab with measurements in the field.
Yes.
How are you doing the measurements in the field?
So the measurements in the field, and I'll come back to that.
Water column from the surface, or what the water sort of close up, or? I'm talking about what sort of instruments were you using?
A spectrometer, a fiber optic. I'll just, you know what?
How was it placed?
I'll jump here quickly to the beginning. I'm using a spectrometer. I'm sorry. I'm using a spectrometer and a light source.
So you're right up close, though.
I'm looking really close here.
Not remote, or not very remote sensing.
Absolutely no. And this is the point I was going to make. Yes. So I'll run quickly. Just a second. Thanks, Colin. No, I'm joking. Yeah, so, we'll get to the last slide now. Okay. So this is actually the point I mentioned, that the next step is making it a more remotely sense approach. And the first step is actually not airborne. It's still keeping it underwater using an ROV or an AUV, something that we can have protected and streaming data when we get close to this kind of a coral head. And now we can sample that and move it around so we know the registration, and we can see the changes that are occurring here, because you have areas that are exposed that are showing coral tips and coral branches. And just moving around, and I'll just make it for the audience to see what I'm talking about, moving around from the tips to the base, looking at these spectral characteristics, I can move to the next level. The Holy Grail is having it from a ship or from an aircraft, but slowly inching it away. However, if you can measure it underwater, you can measure it deep underwater, and then you can move to cold corals. So there's potential going in different directions. We'll still stick in shallow areas, but if you need to dive, you can pop up again quickly. But that's one of the directions we can go with the study. And actually, your question was good also to lead to the concluding remarks here, because the goal of this study is part of a collaboration between IOLR and CCOM. IOLR has a large suite of ecologists that are interested from coral to fish, jellyfish, macroalgae, seagrass, and other kind of species that are occurring in the water column. We have the know-how about technology, acoustic, optic, controls. Merging those two together and having liaisons such as Jen, that you ask her a question about tuna and eelgrass and coral, she has all the answers. She's a great liaison to convey and talk, and if anybody is interested, I'm more than happy to connect you to people at IOLR that are interested in this kind of collaboration. Their main goal, and that's putting there a hint for somebody that's sitting in the back row without saying names, is looking at monitoring commercial marine structures. So now that they have desalination plants and having gas exploration, looking at how to monitor that, and they actually, IOLR has the privilege to go and onto one of his rigs and monitor that and put an ROV that can map along up and down. Actually, they're doing it, measuring salinity and other stuff. That's one of the things that we can utilize for conducting experiments and using it to look in the Gulf of Mexico and other kinds of experiments. So there's other potential here beyond just looking at corals. And to end on this note, I just want to say thank you to everybody, and another kind of research never will happen, but cool. Thank you so much. Yes, Larry.
So following up on Colin's question, you mentioned that the fluorescence is several orders of magnitude less in intensity than the reflectance. And if you're now removed and have to go through the water column, will you still get that fluorescence signal? Do you still have enough intensity at all to see it once you're meters, tens of meters, hundreds of meters away?
Yes, but it depends. So the answer has a caveat to it, of course. Today, you measure from satellite chlorophyll that is in phytoplankton in the oceans. The way you do that is that same spectral fluorescence that I showed before.
Well, that's on the surface.
But that's close to the water surface. It's between the water surface and up to a certain water depth. Here, if we're looking from a ship-borne vessel, so I'm not going airborne at the moment, and you can monitor that fluorescence. Of course, in very high-intensity illumination conditions, which can occur in the summertime in the Red Sea, that might be a challenge. Overcast actually can be a blessing here. Or having artificial light to excite the corals more, such as blue light interacting. Blue and UV light will cause this fluorescence to occur, and that's the benefits of blue and UV. They penetrate quite deep. And corals are quite shallow, in these types of corals are quite shallow, you can see that. So you can remotely, I'm still talking about the order of meters, not hundreds of meters. And the water surface can be an issue, but you can go around it. Tom, you had a question?
Yeah, and maybe I misunderstood something here, but it seemed like it's important to make the contrast between the tips and the base and their different spectral characteristics in order to understand the health. And I guess my question is, as you move far away, as Larry's asking about, do you have the resolution to look at the contrast between those two different pieces of the coral, or is it an integrated measure of both somehow? How do you, if they start to smear together, is it important to separate those?
So, just to make sure that I understand it, when you go remotely, how can you translate an in situ measurement where you're so close you can measure clearly a tip and a base, and remotely where they start to mix together? The nice thing, and this is following Colleen's questions also, when we're looking at it, by definition, tips are typically outwards. So you will get a tip characteristics, and not always you get a base. And not all corals look like this, by the way. There are corals that are much more complex, and that's a whole different story. For this coral, we're planning, and we're still brainstorming, so if you have any ideas, I'm more than happy to hear, but it's actually very similar to backscatter. So after you measure very closely by, zooming out, you have all kinds of features going together and separating it. The nice thing about spectral characteristics, if I know wavelengths, the difference between the two, I can actually put a fraction coefficient there. So I know in this kind of environment, I will have 50% exposure of bases and 50% exposure of tips. So now I'll take a signature, take something that is clear. Let me move to here. Let's say, for example, I'm talking about something like on this kind of situation. This is actually even better. Something like this. So looking at point E, now I have characteristics that's showing an intensity peak at 600 and also, at 580 and lows at 580. So if I can measure that and start to have this density change of lows and highs, I can start characterizing what's going on. If I don't see those highs within that complex image of having bases and tips, then I can assume there's a stress going on there. Yes? Please.
Do you anticipate problems with trying to go further away from the coral in the water, interference with the other algae in the water that might have different pigments or spectral characteristics? Have you thought about that at all? Or
honestly, we haven't.
Way up.
No, but it's a good, and that's actually why we have these seminars, to think about these questions. I haven't thought about it too much, but the benefit is we're trying to avoid chlorophyll as our only indicator, which is one of the common things you have in different phytoplankton. There's going to be scattering. We can't avoid scattering. So light will get diffused. And the farther you get away, you're going to lose it. But if you can keep the resolution small enough that you can identify a colony, a species, that's the maximum resolution we're going to look at. We're not planning to look both of that because then it will be really hard to do kind of characteristics. I think we can do that. But again, there's some research to do here about the scattering due to the size of particulates and also the chlorophyll that is in algae or other things that are floating in the water. Derek?
Yeah, I was kind of confused about what the role of the chlorophyll measurement is. Like, it seemed like the conclusion's midway. Through your talk, you were saying it doesn't seem like they really affect the spectral difference, and then at the end, it seemed like they really did. And I'm kind of confused on.
They actually.
Or I guess my question is, what can you measure? How do you measure chlorophyll, and why would this approach? What additional benefit does it have beyond just measuring chlorophyll?
So first of all, it's a good question because it's a mixed crowd here. Chlorophyll is typically the low-hanging fruit for a lot of vegetation mapping, and anything that has chlorophyll in it, chlorophyll is typically the first solution biologists look for, because you can always fluoresce chlorophyll. And if you fluoresce chlorophyll, you always will see two peaks, 685 and 735. So that's the spectral characteristics of a chlorophyll pigment. Vegetation on land and algae in corals. And this is a conventional indicator. So what I was saying here, we didn't see something that is groundbreaking, that is not repeating the studies of others. That's why I'm saying I don't see anything here I can publish with. But there are changes, as you can see here, from the base tip, from the base to the tip, but the peak here is declining. And the thing I forgot to mention, all these signatures, I normalize them. You get the intensity because you're measuring at different distances. So when we do in situ measurements in the laboratory, it's not exactly in situ. You have to measure with the distance. And these things are not flat. They have this crazy geometry. So I'm always measuring 15 to 20 centimeters away. It's different than actually underwater, where you can get really close and around. Here, we keep all this equipment quite rigid. So I normalize one peak because it's such a stable chlorophyll, and then I can see the changes that are occurring at the second peak, the 735. So there are changes that are occurring there. However, these changes are only an indicator of a chlorophyll. Our goal is to try to map the coral tissue and find out other changes that are occurring in the coral that might indicate that there is some issues going on with these corals due to stress, health, productivity, or some other stuff that's going on. Without, as Jen mentioned, going with traditional biological methods and cutting up and trying to see what's going on, and that literally kills the coral bunch.
So how would you tease apart what's related to effects on the chlorophyll of the coral relative or away from other changes in the tissue?
So for making my life easy, the chlorophyll response is typically above 660 nanometers, as you can see here. The phenomena that where I was indicating were in the green-red zone, which was 525 to 650. So I was looking at a different spectral range on purpose to avoid that kind of situation where we're saying, how do you separate between the two? There was another question here. Val.
Do you have a sense for these kinds of sensors what the integration time is, how long it takes typically to make a measurement? I'm just sort of thinking about what's possible from a moving platform, that kind of stuff.
The integration time can be from a second to milliseconds. It's actually tunable. We tune it according to the light exposure. So actually, when you do a spectral measurement, the first step is you measure the irradiance, the light that is coming down. And based on that is reflected back from a white surface, a white reference it's called, you can actually calculate your integration time because you don't want to saturate your detectors. And then we actually, for these kind of areas, we measure it on 8 milliseconds. If it was dark conditions, like in New England sometimes in the winter, we go almost to half a second integration time. So it can vary, and we can tune it accordingly. The second trick is, of course, having more samples, which you cannot do from our moving vessels. Okay.
Is there like a criteria about the points, the A, B, C, D points along the different genotypes? I mean, they are like spatially distributed.
That was a very good question, and to be bluntly honest, it was random where biologists shouted at me, "Why are you measuring there?" And then I moved it a little aside because I didn't know the difference. So I was putting a point because they were doing like this, if I was doing like that, and so I was moving it. I meant here. So that's typically how it went. So it was tongue in cheek, but it was just areas that I thought, and then the biologist said, "What the heck are you doing? Change it." And it was an iteration. Yes, Andy.
Well, if the biologist could look at the coral and tell you where to take a measurement, there must have been some visual indicator of the coral branch health that so I could swim up and look at it and say, oh, this branch is not healthy or this branch is healthy. If that's the case, why do we bother taking fluorescence and irradiance measurements?
I'll answer, and then Jen can add to that. First of all, it's very hard to swim to a coral and figure out if it's healthy or not. It sounds maybe easy just to go and look at it. One of the things I discovered, there's all kinds of small microorganisms that go on the coral and drill into that and cause all kinds of crazy stuff, which you cannot see just with your eye. You need to take it to see it under a microscope. But in general, just swimming to a coral, unless it's a diseased coral where you can see that, it's very hard to know if you're looking at a healthy or diseased coral. And I'll let Jen add a little more if you have something to say about that.
Yeah, it's really difficult to assess the health of the coral, just visually. And so, I mean, what Shachak's done is really assess the level of stress that a coral is undergoing. And biologists pick those points because those points are areas where there's branching. So they're areas of potential growth. But it's not because they can see that it's healthy or not.
And besides, I understand that the main idea is to make it remote. So you don't have to get close to figure it out how healthy it is.
We're trying to catch on all points. First, helping biologists in the in situ, which is a low-hanging fruit, and then bringing it to the remote sensing, which is our backyard. You have another question, Tom?
Yeah, just along those lines, are you planning to do any sort of larger scale study where you put this in a biologist's hands and they go out and make an assessment of a coral region? Do you have some more field trials planned?
Thanks for asking. And if you can influence the NSF people, yes, I have. So it all depends on NSF, but yes. And NOAA, and NOAA, of course. So, okay. Yes.
So as a representative from NOAA's coral program, Jen is right, it's very hard to determine the health of a coral by looking at it. Even when you monitor it, really what you see is whether it lives or it dies. And so we're trying to figure out sublethal stress measurements. And so what you're working on now is very intriguing. And I'd love to talk to you more offline to figure out how that could get ramped up. So I don't know, I'm just kind of intrigued. It's definitely not my area of expertise, but I'd love to talk a little more.
Fantastic. With pleasure. Yes, Derek.
So it sounds like you did some experiments where you're stressing them intentionally, right? Like
Yes.
in a mesocosm or something. Could you do that with a stress you didn't mention is pollution, like oil pollution? And one of the applications I could see in the future is like stress that's sublethal on deep water corals from oil spills. So it might be interesting to have lab measurements of different levels of stress and different levels of exposure to oil.
That would be cool. If you can grab a sample from so deep and we can have tank. I mean, I'm just thinking out loud, the engineering perspective here, and have tanks that you can put it under the water pressure. But we know what would be a healthy, a controlled environment that we can measure a healthy and then impact that with oil. That would be interesting. Yes, I am interested. I'm just thinking out loud the logistics that are required to do something like that.
You can also try to do that with temperature patterns too.
Yeah.
Because in the deep sea, just like in the pool, I'm assuming they have a fairly narrow tolerance range. And temperatures in the deep sea, at least what they're finding, is that they're very dry.
But the low-hanging fruit is sea temperature. As the sea temperature is rising, we can easily predict what a change of temperature will do on the coral. We can speed it up and see what's going on there and use that. Yes, Susan.
I also think in the area that you're doing your study, you were saying that there's a lot of sedimentation stress.
Mm-hmm.
And so there could be a gradient going from the shore out of sedimentation stress where you could potentially look for changes based on that.
Actually, that's a very good idea. Yes. That's a thing it's worth monitoring before and after one of these floods. Okay. So thank you so much.